So kind of that's true. Mostly I got there a few months ago, so I get to show like the pretty pictures that I've produced. But if you don't like the study designs, I'll give you emails for other people to talk to, because I didn't do that yet. Can't take credit. Okay. So like Jonathan said, we really don't have much of a fundamental ecological theoretical framework for how microbial communities exist indoors, right? We try to apply the way we think about microbes in soils and on plants and things like that to the indoor environment, but sometimes those assumptions are really funny. 90% of our time, on average, in the developed world is spent in buildings. So these are by far the microorganisms that we're coming in contact with most often, and we still don't really know much about their communities, right? We know that they tend to look like humans, but we don't necessarily know why they look like humans, whether we're necessarily producing them or whether we're bringing them in from soils and sidewalks and things like that, right? So at the Biology of the Built Environment Center, we're ecologists and biologists, but we also have architects and engineers that are primarily involved in designing buildings for the future with energy efficiency in mind. And we're trying to understand how architectural choices influence microbial communities inside the built environment. So the architects mainly deal with energy studies, and so any given year in the last few years, somewhere around 40% of our energy in this country goes to just powering buildings. If you break down buildings, somewhere around 40% of that goes to just moving air around, right? So either heating or cooling to make us more comfortable, but also by code, you have to exchange the air every, so many times during an hour. And so since we have to move all this air around inside of buildings, if you can tackle this part of our energy use, this way that buildings operate, you can have a massive influence on the way that we use energy in this country. And that's sort of one of the things that drives a lot of our questions. It's not a mistake we got here. Like it's not very long ago, at least a few key people thought that energy would be free by now. And then while the one correct thing about this is that it's not too much to expect, it's totally wrong. And so we're living with those consequences right now. And so finally, we are starting to design buildings with energy efficiency in mind, not knowing that this is not, that we can't just use whatever we want to. So this is an example of the sort of thing I'll be talking about. This is a middle school in Eugene, Oregon. Some of the architects we work with designed this building. And it takes into account, or it uses natural ventilation. It uses the physics of airflow within a building to try to cut out the fan on the front and cut out the fan on the end and let air flow in a pretty high volume through this building to do what we need to do. So for instance, at night, the building is heated up by the daytime, outside air is cooler, and so if you can open all this ventilation, let air flow through without fans, or there is a fan in this picture, but ignore that one. Let air flow through, that cool air pulls heat off of the mass of the building, and so the next day you spend a lot less energy air conditioning the building, right? And so it's a very simple concept going back to essentially the way we designed buildings 100 years ago before we thought energy was going to be free. But since we're sort of playing with these ways of moving energy around or moving air around, it's important to remember that naturally ventilated systems bring outdoor air in, mechanically ventilated systems move air through heavy filters, through really complex ventilation systems, and as we're doing this, we have a massive influence on the bugs that are moving around in the air and therefore that we come in contact with while we're cruising around in a building. So one of the first things that was done by this center was at a hospital, Providence Health Center in Portland, Oregon. One of the crazy things about Providence, and one of the things that made it useful for this study, is it actually has operable windows. But they had to be pried open for the study because no one had ever actually opened the windows in any of these rooms. By code now, a lot of places, hospitals are built with inoperable windows. You will never be able to open the windows because of this assumption that outdoor air is terrible and indoor air is healthy or sterile, right? Which everyone in this room knows is kind of a wacky assumption. So in this study, they were able to take one wing of the hospital, pry open windows, scrape the paint away, open the windows, shut off the mechanical ventilation system, and let just air circulate from windows, so outside air. The other side of the hospital, which is pretty well cloistered from that airflow, got the mechanical ventilation system, which goes through complex filtering apparatuses, filters that are an order of magnitude finer than what we have, say, in this building by commercial building codes. And so you really get an idea of the bookend of how a really heavily managed airflow system affects microbial communities. In this study, it was kind of overwhelming. The air was drastically different in terms of the communities. In mechanically ventilated rooms, of course it's not sterile actually, it looks a lot more like sick people in their spit. And in the window ventilated rooms, a lot of that stuff gets sort of diluted, even though the cell counts are not drastically different, but you find things that hang out with plants and soils, right? So changes the community without really assigning too much of a value statement to that. It does change the community drastically when you influence the source of air. But hospital, like I said, is sort of one end of that spectrum. It's really heavily managed air, and most commercial buildings aren't like this. And most of us, and maybe this doesn't go for this room, but like most of us don't spend a lot of time in hospitals. And there's not a lot of traffic. There's not a lot of people moving around, stirring up the air. And we know that that is one of the things that influences what is in the air that we breathe, right? But college kids are filthy. And so this building is on the University of Oregon campus. It's some of the architects we work with helped design this building. It's LEED certified, so it takes into account a lot of energy efficiency standards, like passive solar during the winter and active solar to pull in energy, and a lot of natural ventilation schemes to try to cut down on the energy use of fans and let natural air flow through. And so this building has been used because there's a lot of classrooms, there's a lot of traffic, and so in a building where we know that this is designed in such a way that it takes advantage of outdoor air, it's a very different system where air is not heavily handled by machinery. We can actually look at, in this sort of building, the way probably buildings are more likely to be designed in the future, how does human occupancy actually affect this signal, the community of stuff that we breathe. So this is on several different scales. I'll run through a few different data sets. Air sampling was done in these little filtered vacuums for 8-hour blocks, 3 times a day, over a period of 9 days in 2 different months. I'll just talk about one of those months in August. And then over a 9-day period, in the beginning, I didn't do this, they put in clean HVAC filters, took them out at the end of the 9-day period. So these get a huge volume of air that's been traveling in this building and outside air that's been coming in. And we can tell sort of get some proxy for the microbial community that's floating around in the air. And then in terms of sort of a longer time scale, to look at dust that settles, sort of in a geologic context, we're looking at dust in the corners and up on doorways and window sills, and so we used a high throughput filter and pinger device. This one's made by Shopvac, and it worked really well. Let me know if you need the designs. And so basically just sucking dust from all over the building, hundreds of rooms, to try to look at sort of the biogeography of this. I won't talk as much about this. Steve Kemble has a paper coming out about this soon. It's really interesting. So I'll leave that to him. But those 3 ways of collecting data, take a look at the whole building. We get a large scale of a lot of different rooms, a lot of different times, but at one time in one building, in fact, a whole lot of surfaces were swabbed with this funny Whittaker collection plot sort of design, and on walls and floors and desks and chairs to look at sort of geography within a room and how microbial communities differ based on that sort of habitat, if you will, or that contact with humans. So all of these go through a normal process, extracting whole genomic DNA in various ways, steps in triplicate, pulled back together for sequencing. For this, we used MySeq for most of these data. It was paired in reads, and as happens a lot of times with amplicon data, one side didn't work as well. And so most of the data that I'll present today are just using one side. It was a pretty informative region that gave us reasonable OTU distinction, but of course that comes with a million caveats. So on the finest scale, on different surfaces within buildings, since my bosses aren't here, I get to show goofy pictures of them.
We're
on all
posting
these
that
surfaces.
to Twitter, though.
Okay, that's cool. That's totally cool. I think she would appreciate that. So looking at desks, the seats of cloth chairs, walls, and floors. And in fact, these have drastically different microbial communities. They all carry a human-associated signal. They all look somewhat like human skin, but they're drastically different based on where they are, right? So this is like a normal ordination or a classical ordination in that 2 points that are close together have communities that are quite similar, 2 points that are far apart have communities that aren't very similar. But this is constrained ordination, so you can essentially feed it an environmental variable, in this case, which of these categories do you belong to? And in fact, that quite well agrees with these 4 different habitat types, right? So it tends to pull these clouds a little away from each other. If you don't use constrained ordination, it's an excellent way to make your data lie by getting the visualization you want, but in fact, in this case, it's very well backed up by statistics as well. And if you look at sort of these communities on a gradient of a lot of different habitats, you could say that chairs look a little bit more like humans and that the floors look more like plants and soils because that's what we're tracking in on our feet. That is true to some extent, except all of these communities look much more like humans than they do plants, right? And in fact, chairs, this is nasty, chairs, one of the strongest indicators is lactobacillus and a few other gut microbes, right? So a more reasonable way to think about this is that chairs look like butts and floors look like skin, right? Like our skin that we're flaking off as dust. And so they collect very different sets of communities, even though they're all pretty closely associated with us. So HVAC filters in a building like this move a massive amount of air through filters. They're somewhere around 10 to 100 microns. It sort of depends on the commercial coding, but in this case, millions of gallons of air get pushed through filters, and so we can get an idea in the 9 days what sort of things are collecting on these filters. And the thing that really stands out here is, this is just a classical ordination in MDS, but season is really what draws these things apart. They're really hard to compare because of seasonal variation, and then pollen is taken out of this, and so it's not just a pollen signal that would play into it too, but in fact microbial communities are drastically different between the 2 seasons. And so to pull those apart, the same pattern jumps out in that these blue points, which are return air, those are the only filters that saw air that was exclusively coming from a human-occupied space. And bathrooms, arguably the most human-occupied, most trafficked, smallest spaces, right? And so these carry a pretty distinct microbial community than other pieces of air that are coming from return or from supply and a mix of return and supply. So supply air comes from outside through a filter and then gets pushed through the building. And what's interesting, I think, here is that when you mix in return air, you really don't see a whole lot of difference. These communities, even though in some cases they might cluster apart, really, you're still seeing that outside signal, and that differentiates it from the bathroom filters. So the air sampling, I think this is maybe the most interesting part of this project. So these filters were put all over the place in hallways and in classrooms, and this poor person ends up in all of our presentations. in classrooms and outside. And so these were taken a lot of different classrooms, but 9 days, 3 time periods at each day, so 8-hour periods. And these 8-hour periods were 8 to 4, so when there's a lot of people in the building, 4 to midnight when there's fewer, and then the middle of the night when it's pretty darn quiet, right? When things have gotten a chance to settle down. And this idea of night flush, even though there's people in this picture, pretend it's a night class. So louvers are opened up on a hot day when outside air in a place like Eugene is much cooler than it is inside the building because of the hot air that was left over. Cool air gets moved through passively, takes heat off of the mass of the building, and then that air leaves, right? So you're getting a lot of unfiltered outdoor air in a building like this. So this is called night flush, and this is something that I'll bring up a couple more times. Since we have architects and engineers that actually work with this building, we were able to shut this off for some classrooms, leave it on for other classrooms to see how big of a difference it makes when you have air coming from HVAC units versus air coming directly from the outside. So this is another constrained ordination. So the points here are mostly clustered by community on this y-axis. The constrained axis is right here, and it's constrained by occupancy. So the more people there are, we would expect, if those communities agree with occupancy, to get shuffled over to the right. If they agree with low occupancy, to get shuffled over to the left. And you do see that general pattern. It's significant, but that's because there's 200 points here, and so it's pretty easy to get a significant result. But there's a lot of noise here. There's a lot of high occupancy classrooms that don't end up with the other high occupancy classrooms. There's a lot of low occupancy nighttime samples that don't end up with other nighttime samples, right? And one of the things that sticks out here is that night flush plays no part in this pattern, right? They're totally mixed. This is a building that, even though we're opening these vents and allowing a lot of outdoor air, this is a building that takes advantage of a lot of outdoor air. And so that's mixing a lot, and it's not really affecting the community that you see. A short take home from that in terms of the architectural implications, that you can cut energy in half and you don't change the microbial community in the building markedly. And that, I think, is a really interesting result. But back to the noise here. This is pretty noisy. It's not as clean of a result as you'd like to see. And so for kicks, you can throw outdoor samples in here, and those mostly fall in with low occupancy classrooms. And that's not a surprise because there's a lot of outdoor air moving into those at night. But still a lot of noise here. So when you test this day by day, every day, these communities are different, inside versus outside, even though they're pretty close here, and in all 9 days, quite different. However, this is the pattern that I really like. Every single day, these classrooms cluster together in different ways, and that, I think, is really cool. That's a pattern that's not constrained by this ordination, but it's a pattern that falls out even after points are moved around based on occupancy. So the real take-home point is here that the airborne community, even outside, is changing day to day, and that tends to be driving, at least from these data, what's happening inside. So even though outside samples are separate from inside samples every day, for the most part, they tend to track each other as they move around. So there's a lot of shared community between inside and outside, even though a lot of parts of that community are differing day to day. So this is just a single measure of phylogenetic diversity. So the blue got night flushed, the red classrooms did not. No difference whatsoever, right? But if you looked at just indoor air, you would see this pattern and blame that on occupancy or the impending weekend or something crazy happening on Thursday, right? But in fact, when you overlay that with outdoor air, the same pattern exists, and these have the same phylogenetic diversity through the week. So what's happening outside, it turns out, seems to be driving a lot of what's happening inside. And so if you strip this data set down, get rid of the really rare species, and just take those that existed at any given time period, or sorry, more than half of the time periods, either inside or outside, so what we might call sort of the core community in this data set, you find something that looks like this. I did that in the wrong order. So if this is just presence and absence, whether those core taxa are there, by far, most of them are shared. So this is time on the x-axis. Most of them are shared. There's some, and this is sort of the human component here, most of these look like a human microbiome or trace to that. And the outside air actually has a very small component that isn't found inside. So a lot of that in a building like this is making its way inside. This is the same thing, but weighted by abundance. Of course, both of these are funny ways to look at it, but taken together, there is a strong message there. So if we look at the core on either end of that last figure, the ones that are found most often, the strongest indicators inside that aren't found outside, all tend to look like human-associated bugs, right? And the things that are outside, even though some of these aren't totally recognizable, are mostly outdoor environment bugs. No big surprise there. It's good to know that there's not a surprise, I think. But when we break this down, indoor versus outdoor, so these three taxa are things that are commonly associated with our skin. They showed up most often in the data set, or in the indoor part of the data set. And if you look at outdoor versus indoor, these are abundances, so it's hard to see, but I like looking at abundances instead. There's a big difference outdoor versus indoor. These are log values. So there's a big difference in all three of these taxa, outdoor versus indoor. They're more abundant inside. And if you look at occupied classrooms versus unoccupied, the right side panels in each of these, again, very different abundances. That's 20? Wow, okay. I'm moving. Okay, but, so as a factor, occupancy matters a lot. But in fact, if you look at the maximum number of people, right, we would assume that that increases with the number of people. Nothing. There's no trend in any of these. And if you look at the number of churns, this is something that we've come up with to quantify how many classes move in or out during an 8-hour period. There's no pattern there either. And so we came up with this metric to try to quantify how many people are moving in versus how many times this classroom gets turned over. And we went through a lot of iterations of trying to make this make sense in terms of how many people we see, and never does it really matter, right? So as a factor, it matters a lot, and so we've stripped away some of the parameters here, and we've come up with a more simple model, and that is that people are filthy, right? And like, so when 10 people come into a classroom, you stir up a cloud, and that affects the airborne community pretty much just as much as 50 people might affect that airborne community. All right, I'll skip through a lot of this. It's Steve Kimball's back. He's writing the dust paper. It's really interesting in terms of biogeography. Based on human traffic, I'm just going to run some teasers here. When you use the same occupancy load and the same use of classrooms, in fact, you do see a very big difference between naturally ventilated rooms and mechanically ventilated rooms with closed windows. And so only in that context, when you have the same occupancy load, can you start to pick up on that signal. So, to sum up, we're trying to fill in some of the ecological theory of how microbial communities move around inside of buildings and how our architectural choices, since we're redesigning buildings to make them a more energy efficient and a more comfortable place to be, how those choices influence microbial communities inside of buildings. PIs on this are Jessica Green, Brendan Bohannon, and Charlie Brown is the architect. All of these people made samples took samples except for me. I had nothing to do with that. Some of the analysis was done by Steve. Tim O'Connor did a lot of the lab work and was supposed to give this talk, but she couldn't make it. So that's the BioBE. The Sloan Foundation funded pretty much all of this. So thanks to them. And that's it.
Time for a couple questions.
Yes.
So classes take place oftentimes Monday, Wednesday, Friday, Tuesday, Thursday.
Yeah.
Do you see patterns that are associated with people that occupy classrooms?
We've tried and tried to pick that, or I've tried and tried to pick that out, and no, it's really hard to pick up on that. Really, it's the occupancy. I might or might not. I probably don't have an extra figure in here that just talks about occupancy, but occupancy really wasn't that different between those days. Here we go.
But Friday looked different.
So Friday was quite different, right? But ignore the top part. It was just a different iteration of that figure. But Monday, Tuesday, Wednesday, Thursday, and then Monday and Tuesday of the next week, not too different, right? So you're right in a lot of cases. In this building, it didn't matter that much.
So I have a related question to that figure.
Okay.
Convince me that the day of the week pattern isn't some kind of batch effect or something exogenous.
I don't know if I'm going to convince you, but I can say that there are some funny artifacts of people being in these rooms. So this is actually really funny. the funding agent, or the funding agency is Sloan, and our funding officer came to visit, and so there was a lot of activity in this building that day, that night, in that classroom. And that totally shows up in the data set. So actual activities, absolutely. There's definitely some pattern there.
But that you just showed that didn't explain it well in your other plots.
It didn't explain it well in terms of a classroom of 10 people moving in versus a classroom of 50 people moving in. But a group of 30 ecologists scrubbing on walls and carpets and seats and stuff like that apparently has an influence.
They're particularly dirty?
Particularly dirty. Ecologists, yes. Architects, no. I don't know if that's a good answer, but yes. I think the thing that convinces me the most is that we see mostly this same pattern of outdoor diversity, and while that might be that there's fewer students moving around outside Friday, Saturday, and Sunday, I don't know if that, I'm totally convinced by that because a breeze washes most of that out, right? But to me, the difference is a slight change in breeze, and I'm working on pulling in weather data with this. The weather was really constant through the week. Wind did change a little bit, and I think that that's probably going to explain part of this pattern. Maybe there was a, you know, we're close to the ocean, maybe it was coming from that direction versus from the Willamette Valley. I don't really know. So that's a great question, and I can't convince you because I'm not totally convinced myself, but that's what the data say right now.
Just one
I was
more
gonna ask,
question.
wait, you answered that.
Okay.
What effect does student behavior play in this? Because you're looking at students that go into the room, and it doesn't matter between 10 and 50 or whatever. But what about the student behavior outside of their classroom, like hygiene level and that level?
Well, chairs look like butts again. We don't have those data. I have no idea. That I know of, there was never like a drama class in here, so we don't know about like a lot of activity in a classroom. My wife's an actor, so I can say that with impunity. But we did have people sitting in those classes every day, and we do know the schedule. So maybe we could pull out some like sociological artifact there. I don't know if our data are that strong, but it would be really cool if they were. I don't know.
All right, we're going to have to move on. Thank you very much.